Log on, tune in, find out. Another good idea from Cambridge.
Group in the University of Manchester and the BBSRC paid for this project. So in the Whelan group, we're interested in taking data, applying evolutionary models and some forms of statistics, and looking for things that look interesting, where interesting means maybe weird. And I'm invoking the Mr. Men now for the second time in this conference, but typically most people do this, and I'm guilty of this in the past. You start off with a sequence alignment, you make a tree from this, you worry about the tree. Can we statistically say whether we're certain about this tree? You do some statistical tests, whether you're looking for rate heterogeneity or switching rates or positive selection or whatever, and you worry about that, whether your test is justified. But the alignment of the sequences, we just say, well, this is all true, isn't it? But is it? So, in the 25 odd years since Clustal W came out, there've been a whole host of new sequence alignment algorithms and approaches. This is just a smattering of a few, all offering supposedly better alignments. And if they're really better, do they give better trees and better results? And of course, most people ignore this anyway. Most people, if you look at most papers, Clustal W is still the daddy. So why even worry in the first place? Well, thanks to an anonymous Wikipedian for this quote, that Clustal W is used extensively for phylogenetic tree construction, in spite of the author's explicit warning that unedited alignments should not be used in such studies. But yet, everybody does it. And so what we're specifically interested in is if you have a bunch of sequences, so it's a few toy sequences, and then we can try and make some alignments. If we go to the one on the right here, oh, that's good. We've made some assignments of homology here that are potentially not correct. And then we're going to do our kind of statistical test, and we're going to say, oh, this bit looks interesting, right? Because it doesn't fit our kind of null model, because it's not being produced by, well, it's certainly not a Markovian process anyway. We already discussed that in the last talk, but these sites aren't actually related. So there's a bunch of things that people do. Can I get rid of this? Yeah. So one thing is to take G blocks or something and you throw part of the sequence away, and that's a bit unsatisfying because you're throwing potentially the interesting bits away. We can just say, well, we've got a alignment. As long as the alignment's good and the tree's good, then maybe it will work. But prayer might not work because maybe God doesn't exist. We can do statistical alignment. Some people like that. Some people say, well, it takes too long. You don't know when it's finished. Blah blah blah. Or we can manually incorporate biological information, which means you sit down with a biologist and they go, no, it's like this and it's like this. But in the post-genomic era, can we really afford to do that? So what we like out of those options is the statistical alignment, and despite the whinges, it does offer a way of capturing uncertainty over the space of alignment, and also they tend to use explicit phylogenetic models that we know and love, and so we think this probably is going to give the most correct alignments for use in phylogenetics. So what I did was I took 200 sets of sequences from this thing called the Adaptive Evolution Database, on the basis that there might be some adaptive evolution in them. And I threw away anything that had fewer than 10 sequences. So actually I end up with things that have between 10 and 42 genes, but it's quite skewed towards 10. And then we want to know several things. We want to know, when we make sequence alignments under different aligners, how different are they to each other? Then when we go through and make trees from them, how different are the trees that come out? And then when we go and if we do a DNDS test for positive selection, how the set of alignments that are found to be under positive selection, how different are they to each other? And then the actual sites that are found to be under positive selection, how different are those sets to each other? And then because we're doing a statistical alignment, we try and average out across the uncertainty and compare with that. So I took a bunch of standard kind of sequence alignment tools. Sorry if your favorite one isn't here, but really I'm more interested in comparing kind of algorithmic ideologies here. So Clustal is progressive aligner, and then Muscle Maft have a kind of refinement approach on top of that, or at least when you use the defaults in Maft. And then there's consistency-based things. Dieline does something bizarre that I've coloured yellow because I'm not sure what it is. And then the things in red are those that have an explicit model of a tree and then try and do sequence alignment by placing insertions and deletions along that tree. So there's Prank, they're phylogenetically aware, and then Balify is our MCMC statistical alignment tool that we chose to use, and we can take the best thing from that, or we can take some samples from that, or we can try and integrate across the uncertainty. So how do we tell whether two alignments are different to each other and how different they are? Most people actually have what they believe is the truth, and then they just want to look and see how close am I to the truth. That's not a proper metric where you can measure from A to B and B to C and have it obey the triangle inequality and so on. So we produce some metrics that you can do this with. So if you're interested in this, please talk to us. And the nice thing is that our metric, we can also include phylogenetic information and label the gaps based on where they would be inferred to have taken place as an indel along a tree. So if we take our sets of alignments, calculate all the pairwise distances over our 200 alignments, and then squash that all down using multidimensional scaling in a computer, what does that look like? So first of all, the sequence alignments, how different are they? This is what comes out. So the main axis here is about 35% of the variation, and then this one is much less significant. But what we do see is that there is definitely difference between these guys over here, the red ones that are based on the approach of using a tree explicitly, and then we've got the other suspects over here. Whether you use an amino acid model or a codon model in Prank seems to make a large difference as well. And yeah, your approach, the progressive things kind of clustering together here. So then if we then take the alignments and then infer a maximum likelihood phylogenetic tree, what difference does that make? So again, it looks very similar. So one thing to note is that, so this is using the geodesic distance that was discussed yesterday, and this axis here is like 80%, so this is the main thing, and probably you should just ignore this. And what you see is that there really is quite a difference being produced here. So we can kind of, imagine that we can draw blobs around these things and say, look, the statistical alignment here, this is the spread of all the points from Balify, and then the tree that comes out from the best Balify alignment is dab in the middle there. Prank is a little bit outside. When you use the amino acid approach, that's different again. And then over on the other side, we've got all the other alignments and Clustal W out on its own over here. So this is what most people are doing, and it's out on the periphery. And the other thing to note really is that besides phylogenetic and statistical alignment approaches, the model seems to be making a big difference here as well. And whether you view evolution as codons or amino acids seems to be making a difference. So if we ask how much explanatory power there is between going from the distance of between two sequence alignments, the distance between two trees, we can just do this kind of blood spatter plot of everything. But what we do find is that actually there is quite a bit of correlation here. So it seems like if you want to get one tree, use one aligner. If you want to get a different tree, use a different aligner. And actually, it doesn't work so well for Robinson folds because I think branch length is so important. But also, even with the branch score distance, it doesn't seem to correlate anywhere near as well, which is quite interesting. So something else that happens is that when you use a phylogeny-aware aligner, you get shorter trees. So if we plot the total tree length between what comes out of Balify and what comes out of 4 example aligners here, then this dotted line is y equals x, and so we're always above, right? It never goes below. So really, because you're getting shorter alignments and you're squishing together things that aren't squished together in the phylogenetically aware aligners, then you're producing longer branch lengths. And so when we compare the different pranks, so this is the amino acid or the codon approach and the Balify things, we tend to find that the tree length stays the same. So what we want to do is, I don't want to do a load of programming for this. I'd just like to use commodity off-the-shelf software. So how can we adapt using PAML and PAML's M7, M8 test to work on MCMC? I mean, the problem is what you'd really like to do is sample across the M7 model and the M8 model, I suppose, and or well, just the M8 model, and then you look at your parameters and see whether in some Bayesian way they're justified. But nobody does that because it's hard enough to do it without that. So really what we want to do is regard the alignment as a nuisance parameter and say we're not actually interested in the alignment. What we're interested in is our human sequence and where has positive selection taken place along that human sequence. So the usual test, you would have your sequence data, you make an alignment in a tree, you put it in CodeML, and then you get a p-value for the alignment that says whether you reject the M7 hypothesis, which is that there is no positive selection, and accept the M8, and then you also get your empirical Bayes values, which is which sites are under positive selection if we do reject that hypothesis. And ideally, what we would like to do is get rid of that and just have the sequence data and some kind of test and tell us whether there's positive selection. So the way that we've looked at this is to use the marginal likelihood. So we want to get rid of the alignment, right? So then we can compute this, but there are several reasons why we can't really compute this. One is that we don't want to go over all alignments because it takes long enough to do one run of CodeML. The second is that this doesn't really make any sense under the PAML model. Doesn't really have any way of telling me what the probability of a specific alignment is without the sequence, given model M7 in PAML doesn't make any sense. So what we do is we say, well, we're sampling from Balify, and Balify is giving us, we choose random samples from that, so they should all have equal probability. So we just say this is flat, and then we take this from PAML. There's another slight issue that we're optimizing this model M every time. So what you really want to do with the conditional likelihood is have a nice line through across the alignments with the same model and compute the likelihood based across all of this, whereas in fact what we're doing is recomputing M every time, and so hopefully it looks something close, but we don't know, maybe it looks all crazy. But we do have a backup, which is that you can compute this other thing, the profile likelihood, and do the test with that as well, which doesn't integrate across the alignments, but it does allow some variation. So the question of how different are the sets of alignments and the sets of sites under positive selection. So we're integrating across Balify, and then we're going to treat this as our gold standard because we think it's the best we can do. We know it's not the truth, but what is the truth? What is truth? So if we say these are true positives, And so our marginal is our true positives, and we get about 34, I think it is, of the 200 alignments are found to be under positive selection. And if we look across the other aligners, we're getting about 20 to 30 of them, and this is all the samples from Balify. But then if we look at the false positives, so things that are being found across the other aligners, then Prank is coming out as relatively conservative compared to all these things that are finding about 50 other alignments that they're saying have positive selection in. So the question then is this just because these things are hovering around the threshold for Balify, and actually this is just pushing them below the threshold, but we don't think that it is. So if we compute the correlation matrix, so it's the rank correlation of p-values between each aligners, then we get this big red square, which is all the Balify and Prank and so on are consistent with each other. This other red square, which is that the other aligners are all kind of consistent with each other. And then we get this interesting kind of purple line, which is Prank, that is kind of 1 foot in both camps. So to a certain degree, it's whether you do codons, and to a certain degree, it's whether you treat the tree properly. So the other thing is, given our alignments, which sites are under positive selection, and again, we capture most of them with the other aligners. But Prank comes out relatively conservative, and then out of the 350 odd that the Balify was finding, Clustal W finds an extra 1,400. So if you really want to find positive selection, then use Clustal W is not the message of this talk. And the other thing we can kind of plot to look at this and visualize it is the overlap between 3 different aligners here. So I've chosen Clustal W, Prank, and the Balify statistical alignment marginal thing. And what you can see is that this is the most conservative, and it's got reasonably good overlap. Clustal W has got this massive thing that nobody else is finding. So my conclusions are that alignment does actually matter, and we do have to worry about it, which is annoying. And the differences between the aligners. So if we measure the spread from Balify, and then we ask if we take tea coffee and if I take Clustal W, is the difference between the two just reflect the spread that's in Balify? Actually, the difference is greater than that. So Prank, at least on data sets of this size, with all of the caveats, may actually be a reasonable approximation. Looks approximately as conservative as doing a statistical alignment. And actually what I haven't managed to do is all the MCMC with amino acids as well, so I'm trying to do that at the moment to see what the difference is, because it may be that using codons gives additional information, and what would be interesting would be if somebody rewrote muscle or something so that it used codons, but as far as I'm aware, nobody does that. And that's the end of my talk. Thank you. I'll take any questions.
Hi, so I was very interested in your results. The question I have actually has to do with all of your experiments are on amino acids or codon protein coding sequences.
It's protein coding sequences, yeah.
And I don't know if the results are going to be the same when you're not looking at protein coding sequences. In particular, we've looked at lots of alignment methods on larger data sets, but where they weren't necessarily protein coding, and we didn't see the kinds of results you're reporting.
Maybe I'm biased because I'm mostly interested in protein coding sequences. I mean, of course, you can't do the tests that we were looking at on non-protein coding sequences.
Well, you can still look at how similar they are to Balify.
Yeah, okay, you can look at the alignments,
in terms of accuracy, we didn't see the results you're reporting.
In that the other aligners are all quite similar to Balify.
So they were quite different from each other, but it wasn't that the relative ordering you saw in terms of performance wasn't consistent with what you're seeing.
Okay, that's interesting, yeah.
Is there an intuition why the wrong alignments are giving you higher NDS ratios as opposed to just
Yeah, so if you your null model is has fewer parameters, and then you have a model where you're trying to fit extra parameters to the data, and if you just start shunting together things that aren't related, then they're not going to look very good under your null model. But if you've got extra parameters, you can tune it to try and fit that, and you will pass your likelihood ratio test.
We should stop now, go for the break. First of all, please join me in thanking the speakers of this first part of the